What I'd like to talk to you about today are some interesting work that we're doing looking at environmentally friendly polymers, and the objective is to see if we can substitute these for synthetic polymers used in structural materials, coatings, and adhesives. So in nature, if you look in nature, there's an awful lot of organisms that use polymers to make structural materials. Here we have a picture of a sandcastle worm, a reef builder. There's a sea squirt that uses it as a structural material to make the tunic or the body of the organism. And then the oyster, which uses a polymer in making this biomineralized shell. We also have what everyone hopefully recognizes as the blue crab, where they use it to make their exoskeleton and chitosan and polymers. And we also have this winter skate where the egg case is actually made out of a cross-linked polymer as well. And then finally, for adhesives and attachments and holdfasts, we have a kelp here that attaches to the rocky bottom, intertidal bottom. We have the infamous zebra mussel here that uses the polymer as an adhesive on their byssal threads. And its larger cousin right here is the blue mussel, which we'll be talking about in more detail, where it uses the adhesive and a coating on these byssal threads right here. So why is this important? If you look at synthetic polymers and coatings, some if not all require the use of solvents that have something called a high volatile organic component, and the exposure limits that are set by OSHA and EPA make use and application of these materials as costly and time consuming. And also it's not very attractive for dental and orthopedic applications, so there's a push towards looking at natural polymers and coatings. Here you can see images of a spider web silk, or down here a silkworm. And some of these materials have been found to be non-immunogenic, in that it doesn't cause an immune response in a mammalian system. So therefore they have great potential for applications for biomedical situations. So what is the tying theme amongst all this? And it winds up, it's a rather unique and versatile amino acid called L-Dopa, which is shown here, chemical structures here. It's related to something known as dopamine. Most everyone has heard about this. It's used in biomedicine for the treatment of Parkinson's disease. But if we take Dopa and we contain it in a protein, here's a three-dimensional structure, it conveys very unique and properties to the material. Organisms in nature use this unique biochemistry to form adhesives and attachments and protective varnishes and structural materials. And the chemistry that's involved is something called quinone chemistry, where we get cross-linking between these polymers that contain this very unique amino acid. And what makes this possible is that it contains a catechol functional group. And now this is going to be the overlying theme for the rest of the talk, are the involvement of these catechols in natural polymers. It's a very unique chemistry that we find in these organisms. They have the ability to bind metals on metal surfaces or metals in solution. And when involved in quinone tanning, they form some of the most robust biomaterials that are known. Once they're hardened, they're insoluble in water, acids, detergents. So what a great subject to look at as far as an environmentally friendly material. Now let's take an example, adhesion. If you were an organism, why would you have an amino acid like this in your composition? Imagine a world before man-made structures. What would a marine organism have to stick to? Metals and rocks and sand. So here's an image of Amyllis californianus stuck to a rock with their byssal threads here. Can anybody name the top 3 metals in the Earth's crust? Well, there's iron, there's aluminum, and there's silicon. It just so happens that if we look at the affinity for catechols, for metals, those are the top 3 highest binding affinities. So it's not an accident that these organisms have developed the ability to bind metals using this chemistry. Another example is cross-linking. If you were to make a coating, like a varnish or a structural material, what you would really like is to have something that can be cross-linked into a hard, tough material. But maybe you can control that cross-linking so you can make it extensible, depending upon the degree of cross-linking. Over the years, there's been work done where we've looked at the different cross-linking mechanisms. Here, if we have one polymer with a Dopa and another polymer with a lysine amino acid, we can have a Michael type addition here. Not to get into too much organic chemistry, but here's one mechanism for cross-linking. If there's a metal ion, since these catechol bind to metals very strongly, if there's a metal ion present, you have a polymer here, both have Dopas. Pretty soon you can have cross-linking with the polymers, all the way up to having 3 polymers with 3 Dopas binding to this one iron atom. Or you can have Dopas binding to Dopas between adjacent polymers. So here it sets the stage. You've got a natural chemistry that's very unique, and now we want to look at it, see what organisms have these chemistries in nature. This is what's called a phylogenetic tree, listing all the invertebrate organisms all the way up to the chordates back up here. We have beetles up here for just as an example of arthropods. Insects are incredible with this type of chemistry for cross-linking. And then the chordates, we're all related to these guys, as a matter of fact. These are little things called sea squirts. And in their larval stage, they have a backbone. When they come to a surface that they like and they settle down, their backbone goes away and they spend the rest of their life just filtering water. Imagine you're just being able to sit there and just kick cheeseburgers into your mouth all day. Okay, so we're going to talk about 2 specific examples I've been working in the lab on L-Dopa, that unique amino acid, that in 2 different strategies. One is a permanent attachment strategy in coating, exemplified by the blue mussel here. It's where it's used in its adhesive plaque and the byssal threads that it uses to attach to a surface. And then a structural composite material where we look at the oyster, it's a biomineralization and a formation of a ceramic composite. So if we were to summarize what Dopa biopolymers really involve, is the different properties. It contains these catechols, has a high affinity for metals. They adhere and cure in aqueous environments. Once cured, they're insoluble in water, solvents, and acids. They're variable in tensile strength, 99 to 200 megapascals, and that's just for the polymer. That's no inclusion of metal or any mineral phase. Whereas you compare to steel, it's 250 to 700 megapascals. And then you can also make them in thin films, greater than 50 angstroms. Remember, an angstrom is a 1/1000th of an inch. So if you make them very thin, they can be optically transparent to opaque, and they're electrically conductive. So they have potentials as corrosion inhibitors, barrier coatings, new composite materials for applications in environmentally safe corrosion inhibitors, coatings for ships and aircraft, smart coatings, biomedical adhesives, or polymer composites. So here's the organism, the blue mussel. Here's an image of the adhesive point where it sticks to a surface right up here on the glass slide. This is a sequence of the amino acid, but right here is our friend Dopa. Now this section right here, this sequence is repeated about 80 times, and in this it has an open conformation where it can take any, it's basically like a loose string of spaghetti. But what I want to point out to you is that if you take one of these strands, and I've done this just for clarification, okay, if you line it out and just lay one out right next to the other, wherever you see these Dopas line up, that's where you have the possibility for that one type of cross-linking mechanism. That one type of cross-linking mechanism was a Michael type addition, but if you see these blue boxes, that's where you have the potential for these Dopas coming together. Sometimes you're lucky and you can have the choice of either or. Or remember that these things bind metal, so now we can have metal in between the polymer strands, so you can have cross-linking there. And then the rest just goes to show you that it's about 40% one type of cross-link mechanism, 60% the other type cross-linking mechanism. But the whole point of this slide is, if you take these strands of polymer, okay, this one protein that has this Dopa, you can wind up with a very well cross-linked polymer film, almost like an epoxy. In fact, at one point we had taken some of the muscle protein and we had absorbed it onto graphite for 1 second, 15 seconds, and 60 seconds, and we had taken an AFM tip. And an AFM tip is fairly expensive. You can push it down on a surface and generate an image. But we like to use it as a snow plow. And the person that was in charge of the atomic force microscope wasn't very happy when I told him this is what we wanted to do, but he decided to pursue it in the interest of science anyway. And so what we did was we took this tip and we pushed it down as hard as we could, and we plowed back and forth to see how much of the material we could displace, as an example of how much the material had cross-linked. And as a function of time, this is 1 minute, and we could push it all around. If we waited 5 minutes and we tried to push it around, this is what happened. All we could do was just push it to squish it down. We couldn't displace it from the surface. So this is testimony to the fact that this material can cross-link and be very, very rigidly attached to the surface and couldn't be displaced. Which now leads us to, okay, so what? That's all science. What does this really have to do with helping, mankind, business, while maintaining some kind of environmentally friendly status? The Navy was nice enough to give us some money to look at a specific problem that they have. When the ships come into to the dry dock to get repainted, what they'll do is they'll use an ultra-high pressure water jet or grit blasting to remove the paint. Well, it takes a significant amount of time to work your way forward from the stern all the way up to the bow and back again. And what happens is this steel is called a high-strength steel, HY80, and it undergoes something called flash rust. And what happens is as soon as the paint is taken off, it's in a humid environment near the marine coast, and what happens is it starts to corrode. And now if you want to repaint that, now you've got to come back and remove that flash rust. So what the Navy wants is something that they can put into this high-pressure water jet that will stabilize the steel, prevent it from rusting, act as an adhesion promoter for the paint that then you're going to put on. And they want it environmentally friendly, and they want it cheap. So typical of DoD, they want a lot of things for very low cost. So this is what we looked at. We looked at, we took a commercial rust inhibitor and decided to test it against some formulations of this muscle glue protein. So first I'm going to show you a blank is where we've just taken some of the high-strength steel, put it into exposure chamber at 100% relative humidity, 95 degrees Fahrenheit, and from 0 to 30 hours you see that corrosion starts to occur. This is this flash rusting that I'm talking about. The control is where we've taken the buffers that we use the protein to dissolve it in to spray it onto the steel. And so as any good scientist, you want to run controls. And so for 2 days, after 30 days, you can see that rust starts to really take over the surface. At 8 days, at 30 days, we're totally covered with rust. If we take the commercial flash rust inhibitor, and by the way, these conditions that we're exposing the steel to are the conditions that are recommended by the commercial flash rust inhibitor manufacturer. 2 days, 20 days, 30 days, you start to see some corrosion forming here, and then culminating at 30 days. We only ran to 30 days because it really puts a crimp in the graduate student's schedule. If I had them to go 240 days and they had to do 10 experiments, they'd be here forever. So we knocked it down to 30 days, and just to keep that as a constant time period. Okay, so same conditions. Now we're going to look at the muscle protein. All the way 1.5 days, 8 days, 10 days, 30 days. This is a representative slide. We can see that there's really not much corrosion. There's one spot right there that came up at 8 days, but it never got any worse, never grew. And then if we take that protein and then hit it with an enzyme, so we actually enhance the cross-linking, speed it up. Again, we see 1.5, 3, and 30 days. We only get this one spot of corrosion here. So, hey, it looks like it's working. Environmentally friendly material, inhibiting corrosion. If we take the time to visible corrosion and we plot it as a function of treatment, You can see that the longest time is for the enzyme-treated protein, i.e. it's cross-linked. The next is the protein itself, commercial inhibitor, and then the buffer controls. If we look at how much mass loss is one of these things where they talk about how much the corrosion happened. Well, if it corrodes, it's losing metal to rust, the formation of rust. And we can see that the buffer control has the highest amount of mass loss, the enzyme-treated protein and the commercial inhibitor have the next lowest, followed by the protein itself, all indicating that the protein is actually having some kind of inhibitory effect on the corrosion of the steel. And finally, pitting density, which is another nice term to show that we're having some kind of effect on the corrosion on the corrosion process on the steel, with the lowest amounts being for those treated with the protein versus those not, much higher pitting density, in other words, corrosion. So, my graduate student, Will Nelson, is continuing looking at this work for different formulations. The muscle has 5 different Dopa proteins in it, and so he's looking to isolate the other 4 different proteins and look and see how they inhibit corrosion. I'm going to switch gears now. Ceramic coatings, composites. Here's the oyster. What we've been able to determine is that there's blood cells, hemocytes in the oyster, and you can look at how they make this composite material. This is SEM images of the different layers that the oyster makes, but the possible applications are for wherever a ceramic coating is required. Here it is for thermal protection, here's for biomedical surface, here's a ceramic metal composite material. This is an oyster, and this is a section of an oyster shell, and what I want to point out to you here is that you have different layers of ceramic material, and with a polymer integrated into the ceramic. Nice picture here, but you can see these different layers that make up the oyster shell, and here's a nice cross-sectional drawing showing you the different layers. And the nice thing about this, what this demonstrates, is that we have a ceramic polymer composite material. If we take a close-up look, there's something called a chalky layer, and if we blow it up, you can see that it's very porous, ceramic material, similar to what bone tissue can look like. And then you have this dense layer that has a higher proportion of polymer to it. So we're going to look at something where right along here, right along this edge is where new shell is formed in an oyster. This is a freshly shucked oyster. And along here is this tissue called a mantle. But what we found out is that since 1940, the model has been that the shell is formed by this critical supersaturated solution of calcium carbonate precipitating out of solution and going on to this protein matrix, and the crystals deposit on the surface, and that's how the oyster grows its shell. A colleague of ours down at Clemson back in 2004 said, no, that can't be right. After 7 years of research, he caught this on film, where this is with polarized light, and you can see that anywhere where there's these white spots here, these are cells that have calcium carbonate in them, and calcite crystals. And so what they're doing is they're going to the growth margin of the shell and they're depositing the crystals. Imagine you're walking around with a 1 foot by 1 foot square sheet of sheet metal in your gut. And you're walking around, and then all of a sudden you get to the spot where you're going to build a ductwork or something, and all of a sudden you kind of throw up that crystal. Okay? But you haven't killed yourself, you haven't damaged yourself, and now you're able to go back and do something else. That's what these cells are doing. So we took the cells, along with Dr. Hansen here in the biology department, we took the cells out and we put them on metal surfaces, and then we came back after a period of time and came back and looked at them, and what you can see is here's the cell, outline of the cell with some crystals deposited. You zoom in, you can see these areas where very, very small areas where these crystals are agglomerating together. These are lines of where the crystals have been deposited and starting to fuse together. And then if you, this is a piece of 70-75 aluminum, put in an oyster, and you can see this growth front where these ceramic crystals are starting to fuse into plates. And very quickly, we'll look here, we'll zoom in at this interface where we have islands of crystals that have accumulated together. We're going to look at this interface right here. You can start seeing these layers of crystals that are agglomerating together. And remember when I said it was a polymer ceramic composite? Now, if you keep zooming in, higher and higher magnification, you start seeing this stringy, mucousy-like material. This is the polymer. This contains Dopa in it. So, finally, we took I can't remember, this is 316. No, 70-75. So we put a disc of 70-75, put it into an oyster, waited 28 days, then we shucked the oyster and pulled the disc out. And right here you can see the metal disc. Right here is this coating of polymer film. It's like the sheet and the quilt on your bed that's been folded back. So here's the sheet of polymer. If we look on this surface right here, we can see that it's now a ceramic material. So what the oyster has done is put down polymer, ceramic, polymer, ceramic, polymer, ceramic to cover this disc. Here's the growth front for the ceramic part. Then here's a layer of polymer again folded back, and then up here is more ceramic. So here's where it's uniformly coated on the other side of the disc, and then here's where the disc is the coating is fractured. So what we've been able to show is that A, you can take cells out of the oyster, grow a ceramic material on the metal surface, and B, you can do something that's very similar to how the oyster will grow a pearl, and that is you can take a piece of metal, foreign object, put it in the oyster, and then demonstrate how it grows up in multiple layers of polymer, ceramic. And then finally, so this raises a great question. Dr. Barkley and I were sitting talking one day, and he said, how effective is this shell material for blast force protection? And I said, I don't know. So over the Christmas break last year, he took a bag and stood 12 feet away with a 20-gauge shotgun and started blowing the bejesus out of the oyster shells. He came back and he said, I want you to see something. I said, what? And he said, I'm going to hold it on the table. I said, so what? They're blowing up oyster shells. He goes, yeah, but they're in big chunks. They're not powder. They didn't pulverize. So we submitted a proposal to ONR for a damage-tolerant blast force protection material to see if we can get some funding for that, so to engineer a new environmentally friendly material that could be used for armor. So to summarize, biopolymers containing these catecholic functional groups, Dopa, are widespread in nature. We've only touched on a few. They can be used as a basis for developing new organic materials, coatings, primers, and biomineralization mechanisms found in nature can be the basis for a new class of composite biomaterials. And so here's the funding agencies and all the personnel involved.